I'd like to welcome everybody to Gladstone. We're delighted that you're here to share with us some exciting science again. I see many familiar faces in the audience and also many new faces. So particularly for the new folks, welcome if this is your first time to Gladstone. We are celebrating our 40th anniversary this year of Gladstone. So go Gladstone. And Bob and Linda Maley here, who came 40 years ago. Actually, 40 years ago last week was the actual date, are here and have built this amazing thing. So. For those of you who aren't so familiar with this as yet, I'll just very briefly summarize for you who we are and what we do. We are a biomedical research institution. We are independent fiscally and administratively, but enjoy a very close relationship with UCSF across the street, which is an enormous source of strength. But at the same time, we control our destiny because we are independent. So it's really a wonderful relationship on both sides, and it really has resulted in us being able to, I think, be among the top 3 or 4 places in the world in each of the few areas where we focus. So we're not that big. We're about 500 employees, about 25 different laboratories. But those laboratories are organized around a few thematic areas. In particular, neurodegenerative diseases ranging from Alzheimer's to Lou Gehrig's disease to multiple sclerosis, cardiovascular disease ranging from childhood disease to adults with heart failure, and immunologic and viral disorders that you'll hear about more in a moment. I'll say more about that in a second. And we've recognized that science is more and more being driven by changes in technology than it ever has before. And with those technologies come a tremendous amount of data. And so last year we inaugurated a 4th Gladstone Institute around data science and biotechnology, recognizing that that is really going to be the driver of new discovery, integrated with deep knowledge of biology and human disease. And so that is how we're organized, the different laboratories. And we work in each of those areas, in teams of laboratories that are really tackling the big problems in those areas. And that's why I think we've been able to make such an outsized contribution in those areas compared to our relatively small size. We've been able to recruit the very top people in each of the areas and to go at it. Now, in the last year so it's been about 1.5 years since I started as president, and we took the organization through a strategic planning process. Some of you might remember that I mentioned at the last symposium. And we have completed that process and are now in execution phase. And I think we've got a clear path forward, both on the scientific side and on our culture side and our physical plant. And you might have seen that there are some changes in the lobby. So we're actually under construction at the moment. The next time you're here, you'll see a lobby that is much more lounge-like. We've got these interesting ottomans, so you're going to see different lighting. And what we're trying to do is to create more spaces in the building where people can interact in serendipitous manners. And that is because if you look historically at institutions that had unusual periods of creativity and innovation, it's been because of a culture that they've been able to develop where people from multiple different areas happen to come together and interact and ideas bubble up. And so we're creating more and more spaces of that sort within the building for that explicit purpose. So I'm really excited about that and many other areas that we are tackling. So things have been really good in that sense. Now, today, let me just spend a moment framing today's topic, then I'll ask our panelists to come up to the stage and we'll engage in what I think which should be a really exciting discussion. all of you have heard in the news, I'm sure, about epidemics that occur around the world at certain points. There was the Ebola virus epidemic, there was Zika virus, and a number of these pop up and down, and then they go out of the news for a while, and something else comes up. On the backdrop of that, those episodic occurrences, are still infectious diseases that are ravaging populations across the world that you don't hear so much about. Malaria still kills 500,000 people a year, mostly children. HIV, even though it's changed dramatically from a uniformly deadly disease to now a chronic disorder, is still claiming the lives of 700,000 to 800,000 a year worldwide. You don't hear about that so much anymore. TB, you have nearly 2 million people a year die from a TB-related death. So these things are going on, but sometimes they don't affect us in this country, so we don't hear about them. But we think it's critical that we are still focused on this global health issue, where infectious disease remains a major culprit. There have been some major advances in the last few years in cancer biology, where fields have found a way to train the immune system and manipulate the immune system to go throughout the body and find and kill cells. It's a revolution going on right now in that field. One of the areas we'd like to cover today is to think about how we can use not the same, but maybe a somewhat variant paradigm of that, where we can harness our own immune system in creative ways to go find bad bugs in our body and kill them. Can't necessarily do that yet, but there are ways that are emerging given how we might better understand the immune system in the coming years to use it for a whole host of diseases outside of cancer, one major one being infectious disease. And really this ability to train the immune system and understand the immune system has received a lot of recognition of late. The Nobel Prize last year was around discoveries that led to a number of cancer therapeutics. The Lasker Prize, which was just announced last week, which is sort of the almost a precursor, if you will, to the Nobel Prize, was given to the immune system related to discoveries in T cells and B cells, and may talk a bit about that. We have our own Lasker Award winner here, Shinya Yamanaka, who won it some years ago, and that subsequently, he did, of course, receive the Nobel Prize for that, as many Lasker recipients do. So this is... This is just to illustrate the interest currently around the immune system and how one might be able to harness that for a variety of diseases. And today's topic then, of course, will focus on the immune system. And before I invite our panelists, I just wanted to also mention that this symposium is sponsored by our President's Council. And for those of you who don't know what that is, this is a core group of supporters that have been engaged with us for many years and have really been critical to both supporting the science here, but also sharing our stories with others and their friends, and spreading the word about what wonderful things are happening here. So I thank the President's Council and their leaders. Will Evers is here, Fred Dorey and Laura Dorman serve as leadership for this group of about 100 or so really dedicated supporters. So thank you all for doing this and for supporting us. So at this time, let me ask our panelists, Warner Greene, Melanie Ott, and Don Ganem, to come up to the stage. And I'll briefly introduce them when they're up here and have them introduce themselves. Their bios are in your programs, and I will serve as moderator for this discussion. So Melanie Ott is one of our faculty here at Gladstone and leads a program called BioFulcrum, which she'll tell you more about. Warner Greene was the founding director of the Gladstone Institute of Virology and Immunology in 1991, and really was instrumental in this charge worldwide to make discoveries and lead to this change that occurred in how the natural history of HIV, which is remarkable, and he directs our Center for HIV Cures here. And Don has been a faculty member here at UCSF some years ago, and then went to lead, be global head, you see in your notebook notes about with Novartis in this exact area. And so he's going to be able to share a unique perspective of both academia and industry and the interface of that. So with that, I'd like to maybe, Melanie, if you could start and maybe just say a few words about yourself and your interest, and any comments you want to make introductory, and then we'll go into some discussion after that.
Yes, welcome. Thank you for coming. I'm on the panel here as a virologist, but in fact, that's not how I set out in, in medicine and in biology. I was a clinician in Germany in the beginning of the 90s. I was a chief resident in neurology of an intensive care unit, and at that time, I had 90% of my patients come in gravely ill and dying, and they were all infected with HIV. That was really a terrifying time, and I think all of us who know that time will remember. There was very little we could do at that time, and it made me change courses. It really affected me so deeply that I moved to New York, started a PhD in virology because I wanted to get to the bottom of what that virus was doing to my patients and to so many other people around the world. And that got me hooked, and I've never gone back to clinical work, and I have, ever since studied viruses and how they affect our body. And the reason why I got hooked is because viruses are really fascinating. Viruses show us the way to amazing biology in the body. Deepak mentioned the immune system. I mean, the immune system has evolved to deal with viruses and with pathogens. So by following viruses into the body, we really discover amazing biology. And it is the good and the bad in our body that we discover. We discover how our body is throwing lifelines to viruses, for example, to survive in our body, because the virus cannot survive by itself. It needs critically our help. But it also shows us what we do good and what we have evolved well to deal with and for example, defend against the pathogens and the viruses. So as Deepak said, we have started recently here at Gladstone a new initiative, which is really systematically studying these interactions between the pathogen and our body. So that we can develop new therapies that are not going after the pathogens, but they're going after these reactions that they elicit in us, either strengthening the immune responses or cutting off these lifelines that we are going to throw to the viruses. And we think this is a new approach. The regular way of doing this is, for the longest time, was really developing antibiotics against bacteria or antivirals against viruses. And this is a very successful, strategy that has led us to, fantastic HIV medication and hepatitis C drugs, but they come with a price. And the price is that the viruses are smart. They can develop resistance in very short time. And it leads to us requiring 3 drugs against 1 virus just to avoid that they become resistant. So our goal is to develop these drugs against, our own reaction to the virus. And we hope that we will actually require potentially 1 drug against multiple viruses or pathogens. And with this, I give it to Juan.
Thank you, Melanie. Let me offer my thanks for your attendance today as well. So I am trained as a basic scientist. I have spent my career doing fundamental research around HIV. But what I wanted to relate to you is a story in 2007. I really didn't fully understand HIV until I made my first trip to Africa. And I did so as becoming the president of the Accordia Global Health Foundation, succeeding a very good friend of mine and a friend to all of us, Merle Sandy, in that role. The Accordia Global Health Foundation was dedicated to curbing the impact of infectious disease in sub-Saharan Africa through the creation of African-owned and African-led centers of excellence. And the first center that we helped create at Makerere University in Kampala, Uganda, was the Infectious Diseases Institute. And this was the first trip for the inauguration of the Institute, which was largely financed by Pfizer. And I remember walking into the clinic, this clinic about the size of this room on a Monday, and it was full of people, about 300 people, and you could hear a pin drop. And I thought to myself, wow, it's really quiet here. But then my guide and I happened to be in the clinic the next day, a Tuesday. And the clinic was also full, but now everyone was chattering, kids running around. It was just an amazing contrast. And so I asked my guide, I said, what's the difference? Oh, he said, today, Tuesday, all of these individuals are participating in a clinical trial and are receiving antiretroviral therapy for their HIV infection. They're getting better. Monday, they're just coming in for checkups. Everyone's dying. So I wrote kind of a trip report, and I entitled it, Turning Mondays into Tuesdays. And I'm delighted to say that the Infectious Diseases Institute, which Accordia was really blessed to have had a major role in its formation and success, is now, for example, caring for over 100,000 HIV-infected people. Its outreach programs in the rural Uganda have reached over 2.5 million people. They've performed 250,000 circumcisions to prevent the spread of HIV infection. They've published 500 papers. They've trained PhDs, graduated from their programs. They even opened recently a new bioinformatics center. So, and they're a clinical trial unit. I couldn't be more proud of the activities, and I think in the long run, it is this kind of building capacity, building healthcare capacity in country is really going to make the difference when the next epidemic comes along. Not HIV, but whatever the next epidemic may be. We want to make sure that we've built strong health systems in the most vulnerable countries to withstand the burden of that disease.
Thanks, Warner, and thanks to you, Deepak, for inviting me and all of you for showing up. I don't have nearly as dramatic a life story to tell as my two colleagues here. I'm just a middle-class white boy from Massachusetts who has been interested in science since he was a sophomore in high school. So I won't regale you with any of those, any of the interceding events. When I got to UCSF, I was completing my training as an infectious disease specialist and virologist, and spent the next 15 years working on hepatitis B replication and pathogenesis. As that program matured, I was part of our infectious disease service here during the, I was a fellow in 1981 when we saw the first case of HIV in San Francisco. And then there followed this extraordinary decade where to practice medicine in San Francisco then was like practicing medicine in Africa now. Untreated, the full force of the untreated HIV epidemic was here in San Francisco. And I got extremely interested in that. I never wound up working on HIV itself, but got very interested in Kaposi sarcoma, the neoplasm that was at that time the #1 tumor of untreated HIV patients, and went on to work for the next 15 years on the herpes virus that we now know causes that disease. And that was a very pivotal thing for me because it was the first time in my career that I was doing clinical investigation at the same time I was doing laboratory science. The reason that came about was the virus had just been discovered, viral genome had just been discovered. Actually, not even the whole genome, just 150 base pairs of it had been discovered. And but it was clear that one had a handle now on what might be causing KS, but I didn't want to be spending the next 10 years working on a virus that turned out to be irrelevant to KS. So we realized that at that point, we had to study patients with the disease and establish epidemiologically that this genome was in fact coming from the virus that was the etiologic agent of KS. And I'm proud of the fact that we were one of 2 or 3 labs that actually established that this was in fact the causal agent of KS. And then we spent 15 years working on all the different mechanisms by which that that was exerted. So that was my story of how I came to be here. That experience in working on KS and doing clinical investigation as well as science led to another trajectory of mine that was unusual. Most physician scientists stop doing medicine and spend more of their time working in the lab. As a result of that experience, I was so exhilarated by clinical investigation that I spent more time attending in medicine. By the time I left here, I was doing 2 or 3 months of attending in ID and got really committed to the idea that I wanted to spend the next part of my career not just contributing to microbiology, but contributing to medicine and the treatment of infectious disease. So in 2011, I left UCSF after 30 years to become the head of the infectious disease development program at Novartis, where I spent 8 years until now.
Well, thank you, guys. Maybe we'll start off, maybe Warner, you have some really interesting work coming out soon that'll be published that I wonder if it might be interesting to share with the group on, in fact, what we talked about, harnessing the immune system for HIV. Maybe you could share some details about that.
So I'm sure that many of you have heard about these new CAR T cells, which stands for chimeric antigen receptor T cells. Essentially what you're doing is reprogramming the T cell to attack a specific target. And in many cases, it's a tumor antigen, a tumor marker, and it's been highly successful in many types of leukemia, for example. We partnered with a company that was involved in CAR T cell, but it's a different type of CAR. It's a convertible CAR. And the idea was that we could now attach any antibody. We would target this killer cell, but we would target it not with inserting an entire antigen receptor into the cell, but rather we would do so with existing antibodies. So we could modify the antibody so it would latch on to the cell in a very, very specific manner. And we took advantage of the fact that there are now broadly neutralizing antibodies against HIV, and that they interact with the envelope protein. And the idea was to try and attack the latent reservoir on the way toward a cure. So one of the most popular approaches for an HIV cure is to try and shock the virus out of its quiescent state and then kill those cells, make sure those cells are dead so you can reduce the size of the reservoir. So this is the kill.
Maybe, Warner, it might be useful for those who aren't familiar with the latency issue of HIV, which is a key issue of why we can't cure it today.
Sorry.
It might be good to mention.
Sorry, I live in the world of latency, so I apologize. And Deepak so nicely, so latency is the reason that we have to give antiretroviral therapy for someone has to take it every day for the rest of their life. Because if you stop the antiretroviral therapy, there is this latent antiretroviral therapy resistant virus that can now spring out and reseed the entire infection, and you go back to having full-fledged HIV. So the idea is, so viruses in general persist in two ways. They either continue to actively replicate, or they establish latency and then come out intermittently. HIV does both. And so this latent reservoir is the real vexing problem of why we don't have a cure, why now we have to treat 37.9 million people worldwide. And frankly, 62% of the people, we now have antiretroviral therapy to 62% of those people. It's an amazing accomplishment. And George Bush gets huge credit for the initiation of the PEPFAR program, which will save hundreds of millions of lives ultimately. But back to the CAR T cells. So now we have to trigger the virus to come out of its slumbering state. And once we can do that, then it expresses these antigens on the surface, these proteins on the surface, and now we can, with our broadly neutralizing antibody attached to the killer cell, we can retask that killer cell to go and kill those activated reservoir cells. So it, it's kind of Star Wars. It's kind of, but it works. Now the problem is that this therapy costs about $500,000. So you think about it, you have to take the cells out of the person, you have to re-engineer them, and then you have to infuse them. So this approach, this is really a proof of concept approach. It really will only become practical in the setting of HIV when we get a universal donor cell that can all you have to do is mix the cells that are on the shelf with the antibody and infuse. That would be doable. But we need these universal donor cells. But at any rate, the CAR T cells may have an application ultimately one day in infectious disease, just as they're impacting the world of cancer.
Thank you, Warner. So in cancer, this is one of the methods I mentioned of training the immune system to go find and kill cancer cells, and CAR T is the approach. Because of the deep interest in cancer, there's a huge amount of private sector investment in this technology, including in the efforts to make a universal donor. So right now it's so expensive because you have to take a person's own cells out, engineer them, put them back in. But if you had an off-the-shelf product that could be administered to anybody, then it just changes the game. And I'm hopeful because there's so much investment in that world because of cancer that I think will then quickly translate over to this. So that gives me hope that it could be viable. But ideally, it'll be some other approach that is not so expensive. Even that's going to be pretty expensive, cell-based therapy across the world. Now, Melanie, I know you have an interest in antibody engineering and potentially using that. So while we're on the topic of antibodies that can go and find and kill things, it might be interesting to hear your thoughts about that.
So you heard all about T cells, which is one part of the. Is that on? Here.
And maybe to start this off, it might be good to give a primer for those who last took high school biology and learned about B cells and T cells. If there are any of you out there of that nature.
We will try to keep it very light, probably lighter than in the high school. But the point is not to stop just in one cell type, T cells. And by the way, CAR T cells were actually developed not for cancer, but for HIV originally, and then have made a huge success story in cancer and now are coming back to HIV thanks to, Warner's work and others. So the idea now that we are following is that not to go after the T cells, but to go after the other group of cells, which are the B cells, or even after non-immune cells to give them some immunological properties. And if you imagine if you get a vaccination, that's what usually is triggering your antibodies to be produced from your B cells, and that gives you protection. It can also be used as a treatment, and we know, for example, from HIV, but also recent successes from Ebola, that just taking these antibodies that are very, very efficient, and these are the broadly neutralizing antibodies that Warner was mentioning, they really work, they kill the pathogen, and they're not just tickling it, but they really are effective. So these broadly neutralizing antibodies are the ones that we want to elicit in everybody, as a prevention or as a treatment, but it doesn't work. For example, for HIV or for hepatitis C, we still don't have a vaccine that would effectively do this. And so the idea is now what we're doing is we isolating or producing these antibodies in a factory. So you can make them synthetically and you can produce them and you can infuse them. But you can imagine that this is not going to last for a lifetime. This is just lasting for a few months. And then you have to redo it and redo it and redo it. And it's not necessarily becoming easier to reinfuse them because the body develops all kinds of reactions against it. So the idea is that you now take the antibody and instead of infusing it from the outside, you engineer the B cells or even the muscle cells to produce these antibodies. And with, Jennifer Doudna, who recently joined the Gladstone for a certain effort, and her CRISPR technology, we now have really, unprecedented abilities to engineer our genome and to take, for example, a gene of an antibody and to modify, and to put it into the genome of our B cells or into the genome of our muscle. And the B cells are particularly attractive because the B cells already have antibody genes, and so you can just make minor tweaks to make the antibodies that people might already be producing to be much more efficient. But that again goes into the direction that we have to take the B cells out, we have to engineer them, we have to reinfuse them. So that's a huge cost factor unless we get universal donor cells. If we go into something like a muscle, the muscle is not as good as a as a B cell to make proper antibodies, but it's pretty good, and we know this from from previous approaches. So the idea is that we might be able to actually do an injection of something that would engineer the right type of antibodies into our muscle cells, so that the muscle would continuously produce the right antibodies and would protect us, or even treat treatments if we are already treat the diseases if we are already infected. And I think this would be a much more cost-effective approach, and would be something that is now becoming possible due to the progress that we made with CRISPR biology and close collaborations that we have with Jennifer's lab.
Thank you, Melanie. Yes, this is an example of what I raised earlier about technology. So the CRISPR technology, for those of you who are not familiar with it, is a very efficient way to do gene editing and manipulate the genome in a cell. And Jennifer Doudna is the co-inventor of that technology, has a laboratory at Berkeley, and last year wanted to set up a second laboratory in a more biomedically research-oriented environment. And so she elected to do that here at the Gladstone. And so her lab is now interfacing with many others, bringing that technology to various aspects of disease biology, like what you heard from Melanie, that's those kinds of things that are really driving the pace. Where here you might be able to go from a really expensive treatment to something that would be essentially a gene therapy, introducing a gene into a muscle and then letting your own body do the trick and make what you need to battle disease. So that's, I think, a really good example of that. Don, I know, given your experience in academia and pharma, you've thought a lot about the kind of approaches we might be taking in academia, and now thinking about, can these really become therapeutics, and all the challenges that ensue in between there. So I think it'd be great for the audience to hear your experience having gone to pharma and seen it from that side, about how do you actually take some of these discoveries and get them to people.
Well, it's a mixed bag. This is a complicated subject, and we're living in a time when I think there's a lot of policy making around academic institutions getting more involved in applications, and I have mixed feelings about that. When I think back to my experience in the industry and look at the industry as a whole, there's one thing that's very clear. Every single thing we were working on at Novartis in terms of antiviral development, for example, was the result of progress that had been made in academic virology over the 30 years that I've been in the field. If you look, for example, at influenza, there are 2 new drugs for influenza that are working their way through. One's already been approved by the FDA. One is in phase 3 now. These are drugs that target 2 subunits of the influenza RNA polymerase that are involved in binding to host messenger RNA and stealing the caps off of host messenger RNA to use to prime viral transcription. I remember when the paper that described that whole pathway, which is absolutely unprecedented, was published in the early 1980s by Bob Krug, who's now at the University of Texas. He actually just wrote to me on his 80th birthday, and I told him he was unaware of the fact that there's a second molecule on this pathway that is working its way through the regulatory path. But that is an example of what academic science offers industry, is fundamental discovery that shows us new pathways that can be inhibited. That is that is what academicians do best. I don't think academicians are in the strongest position to take that discovery and turn it into a small molecule inhibitor of the pathway. That requires a bunch of things that academics was not set up to do, okay? First of all, that quest from a pathway to a drug is a 20-year quest in general. Some would say it's even a 30-year quest. If you look, my old colleague Mark Fishman did a review of fundamental discoveries and how long it took for them to become drugs. And it's his view that it's more like 30 years, okay, from the initial discovery of the pathway to the drug. And he used statins as a good example of that. However you slice it, it's a long time. If you think about the way academic science is structured, everybody's lab, academic science operates on a cottage industry model. The cottage is the individual PI. The workers in the cottage are graduate students and postdocs. Those graduate students are in graduate school for 5.5 years, postdocs for 4 to 5 years. Each one of them needs to have a project that he or she can call their own and that they can turn into a publication that is then becomes part of the public knowledge. The entire system of academic science is not set up for a dedicated sort of Bolshevik level quest of producing a product in 20 or 30 years. In fact, it's set up to do the opposite, to disseminate knowledge that is then part of the public domain, can no longer be patented. So I think it's, and what disturbs me now is we're seeing a lot of policy being promulgated at the federal level, trying to get academic institutes to do more of these sort of applied work and less of what the system was built to do, which is discovery work. And to me, it's the complementarity between what industry does best and what academicians do best that is responsible for this huge output of the past 30 years. And that we disrupt that ecologic system at our peril. I see a lot of academicians who are being encouraged to do high throughput screening. Now that robotics has become cheap enough for every university to have a medium throughput capacity, the most popular experiment that in academic science now is to take the lexicon of FDA approved drugs and screen it against some new target. That has almost never resulted in a new important advance. You take drugs that have been optimized to have a 30 nanomolar IC50 at a particular compound, you screen them at 10 micromolar, and you discover things that are very weak inhibitors that are totally have nothing to do with the purpose that the drug was originally developed for. Very rarely does that result in a drug. So I think, the kind of things that the Gladstone has exemplified over its 40-year history here is exactly what academic science should do, is make it possible to think about the Star Wars applications that have just been mentioned by Melanie and Warner. Those would have been inconceivable a long time, but they're still probably 20 years away from actually happening. The universal donor cell, how do we get there? Well, now we can start to think about how one gets there. CRISPR out all the MHC genes, think about things that would make the cell not be rejected by the immune system. But the ability to even have that thought is the product of 30 years of academic immunology. So, we're a long way from these things becoming imminent therapies, but the ability to entertain them is what I find exciting. And that's the product of the last 30 years of academic work in virology and immunology. Could the progress be faster than that? Yeah, maybe it will be, considering the amount of private sector investment going into immuno-oncology now. It might be faster than another 20 or 30 years, but we shouldn't count on that. We should be, and I think we shouldn't make policy that redirects academic science in a direction it was not structured to do because of a mistaken belief that the things that academic science did do well that led to drugs means that they could actually do the whole enterprise themselves. I don't think that's true. I think maintaining this complementary structure between what academicians do and what industry does is a better formula, and we rely on academicians to have ideas that are bold enough to embolden industry to turn them into a practical therapy. That, to me, is a better way to do business.
Yeah. And I think at Gladstone, we try to walk that line where our sweet spot is discovery, biology around normal and disease states. We stay in our sweet spot, but when things might have potential to turn into a therapeutic, I think the difference, Don, is we've tried to be active in partnerships, creating partnerships, rather than putting it on the shelf and hoping that somebody identifies it and takes the next step. While we stay in our sweet spot of discovery, we've been proactive about bringing the right partners in to take it to the next step. But I think you're absolutely right. It would be a mistake for us to then think that we could do it at the same level of expertise as people who know how to make drugs.
I just want to regress for a moment and talk about Ebola. We all remember the West African 10,000, 12,000 people infected, and about a 50 to 60% mortality. And now we're in the midst of the second largest Ebola outbreak in the DRC, Democratic Republic of the Congo. 3,000 have died. But there has been a miracle that's happened with this disease. Melanie kind of touched upon it. The NIH launched into trying to come up with a new therapeutic. Regeneron also, using two different approaches, but both came up with monoclonal antibodies. Antibodies are becoming very good drugs that can attack the Ebola virus, and if given to people early in the course of disease, can be curative. Early in the course of the Regeneron product, mortality rates were dropping from 79% to 5%. I mean, we never have seen anything. Ebola is a horrible, horrible virus. But there's another thing that's happened, and that is there's now an Ebola vaccine that can be used in what's called ring vaccination, so that you give the vaccine to anyone who's even come close to this person. And that's now having a 97% protection. So here's an example of an absolutely lethal infectious disease through the combination of basic science, rapidly translated into antibody cocktails and into a vaccine, has led to a very different and very pleasing, I mean, very optimistic that we're going to be able to control this virus in a way that has never been possible before.
Melanie, you were going to...
Yeah, no, I can only support this. I think the interesting thing is that these antibodies were originally identified from survivors, very rare survivors. And what happened in the last epidemic before this one, we were using serum from people who survived and infused, not we, but people in the field used the serum, and because that contains the antibody, and infused it into patients, and that had an effect. And within, the time span of these two epidemics, people were able to isolate the effective antibodies, and were able to produce them at a large scale, so that for this epidemic now we have the right antibodies ready and can give them to patients very early in the disease. The key is really for people to come early, and that remains a big problem because it's still a stigma, and people don't want to, be exposed to being isolated and, having their whole houses taken apart and being separated from their families. So I think now, since we have this fantastic success, I think we have to get the social environment to catch up with it.
Yeah. So, Don, you, I know have thought about the fact that given the interest and investment that we talked about earlier in immuno-oncology and many other areas, that drug companies are often finding it more attractive to invest in those areas. We struggle with the same thing in the Alzheimer's space, where many drug companies, it's hard, it's difficult stuff, so people have, many pharma are exiting that area. It's been true for heart disease for a quarter century. There are very few new drugs. They'll often find the path of least resistance to try to develop drugs in those areas, which makes sense. It's a logical business decision. So what, my hope is that if the paradigm shifts from some of the technological advances that we're talking about and many others that'll make it more attractive, because there's still a huge unmet need in this space, but the paradigm has to change. Do you see that happening in the near future, or what would it take, I guess, is the question, for that to happen?
Yeah. So let's be clear about what the problem is. Most of the big pharma companies have gotten out of the entire infectious disease sector, not only including drugs for the developing world, but including antibiotics and antivirals for the developed world. Now, why is that? It's not that these drugs aren't profitable at some level. It's that they're not as profitable as therapies for cancer and autoimmunity, where the marketplace has shown that it will support extreme premium pricing. And so now infectious diseases are actually more prevalent than a lot of the cancers that are out there. So you'd have thought the market would be big enough to support this. But let me make the analogy to the food market. You can go to Safeway, and Safeway can charge you $28 a pound for filet mignon or Atlantic salmon, because that's a delicacy, and you don't eat that every day. But you can't charge $28 for a loaf of bread or a head of lettuce. Antibiotics and many of the antivirals would correspond to staples of medical practice. And even though the number of people who are needing them is very large, it's precisely because of that region that you can't support premium pricing for those compounds. So these are small, low-margin drugs, low-margin, high-volume drugs. And the current financial system that we have in this country is rewarding high-margin, less frequently prescribed drugs for a whole variety of reasons. So if you're the CFO of a major pharma company, you face what's called the opportunity cost issue. Every dollar you spend on an infectious disease program is a dollar you're not spending on a more remunerative program in oncology, immuno-oncology, autoimmunity. And that's a serious problem. Therefore, most of these companies have gotten out of that business. And the net result of that is, well, people say, well, gee, well, maybe small biotechs could step in and take over this space. I've heard that argument, but when you look around at the universe of small companies that have tried to make antibiotics, almost all of them have gone out of business. Why? Because they can't sell the product to a big pharma that's capable of worldwide manufacture and distribution of them. None of them want to do that anymore when these other products are more remunerative. So we're facing a real fundamental problem here that is ultimately traceable to the over-financialization of our economy. The fact that Wall Street pressures these firms to make these decisions. And if you live in a culture where the only important value for a publicly held company is maximization of shareholder value, if that's the only objective, then this is the outcome that is the ineluctable result of that dynamic. Now, the good news is that we're starting to see a shift in the attitudes of investors about that. You may have seen recently that there was a big powwow on Wall Street that said maybe we should rethink the idea that the only objective of a publicly held company is the enhancement of shareholder value. If that now is at the level of voluntarism, everybody's making rosy public statements about it, but it isn't a matter of public policy. It isn't a matter of American tax policy. It isn't a matter of American Medicare reimbursement policy yet. But we're starting to see on the political stage a lot of change about that. And if those ideas change, and the idea that maximization of shareholder value is not the only thing that a big public company does, then we'll see change here. But I don't see this situation auto-correcting itself. It's going to require the political system and the tax structure to change in order for that to happen. And I worry that it might take an epidemic of a multi-resistant Candida auris, which was on the front page of the New York Times today, or multi-resistant, gram-negative infection or multi-resistant tuberculosis to rectify this situation, because, I don't see the current dynamics of the marketplace altering themselves in this direction, and I hate to be downbeat today. But let's talk about the flip side of that, which is it creates a greater imperative for discovery in academic science. If new targets are not going to be explored in the industrial setting, it creates an opportunity for new thinking about this in the academic setting to possibly make a new approach possible that would lower the cost of goods and make a new approach possible. So I think there is an upside for institutes like this to stay invested in this area and to continue to do discovery research. But ultimately, I don't see Anglo-American capitalism voluntarily changing course here. I think there needs to be a push to change the market incentive.
We are in a time where, because of some of the new approaches and technologies that you heard, whether it's for infectious disease or other areas, we're starting with a foundation now where I'd argue over the last 30 years in all fields, the scientific community collectively has been building the knowledge base to the point where we are ready to launch. And I would hope that while in the past some discoveries took 30 years to get to a product, it's because they had to do so much more foundational work. We're now starting with a base of foundational knowledge and overlaying on that tools and technologies that we couldn't even dream about a few years ago, particularly for human disease, which we didn't really have before. And so it's still going to take a long time to go from target to a therapeutic, but I've never been more excited about the prospects to tackle so many different human diseases than what we have today, and particularly for what we talked about today, infectious diseases. I think the key is for places like ours, particularly given the landscape that Don illustrated, that places like ours to remain committed to this and innovate our way out of the problem. The capital markets may or may not change. The pharma math may or may not change. I think the main thing we can change is the paradigm, and that's on us to innovate out of this conundrum, and I have a lot of faith that we can do that. And in fact, at Gladstone, in this space, we are getting ready to recognizing that HIV, while not cured, is a very different disease than it was when we initiated our effort 28 years ago. We are now trying to take our same Gladstone model that's been repeatedly successful and pivot it to how we could harness the immune system for infectious disease and other areas. And we will be embarking on a recruitment of 4 new faculty in this area in the coming years, which will really shape, we think, how we can tackle this problem. And 4 faculty in our system is a lot and can make a huge impact, we know, worldwide on disease. And so one of the things we'll be coming to the President's Council with is how you all can help us raise the funds to tackle this problem and bring in new faculty, new talent. And so what I'd ask of all of you from today is share what you've heard here with your network, with your friends. Some of them may want to get engaged with us in various ways, and some may end up being friends of Gladstone and communicate to their friends. Some may even become supporters of our science financially, and that's the kind of thing that really will fuel us going forward and allow us to achieve these goals. But we welcome you to partner with us in this, what really is a noble mission to conquer some of these still devastating diseases. So thank you for joining us today, and please join us for some cocktails and food outside. Thank you to our panelists.